Wonderful. So, and just to correct the record, I know Nino, I don't know if he's left, but his title actually came from Savannah. So that was not my idea. In fact, I think most of the titles here came from Savannah. So my title that was assigned to me was High Throughput Modeling for SIDS Risk Factors. Just to get things started, no conflicts yet, as I always make that joke, because we are hoping to one day commercialize this in the future, but just spread that out throughout the community. I do want to give a sensitivity warning. Of course, there will be discussion and imagery of deceased infants and animal experiments ending in death, and I will show a live video of that. Not them dying, but of live animal experiments. And just so that we're all on the same page, sudden, unexpected, and unexplained death of an infant is referred to differently across the world in various agencies. So for brevity, I will be using sudden infant death syndrome, but depending on the context, this could mean different things. Okay, so I wanted to get on my soapbox a little bit before I get into the science and share some things about this problem that I think are quite prescient and important for all of us to understand as we try to solve this. I think we can all appreciate that all aspects of sudden unexpected death in pediatrics are a serious issue in the United States and around the world. It is an outsized unmet medical need here. So while we have reduced SIDS rates with the Back to Sleep campaign, they have plateaued and they're stagnant. We are not reducing these further, and we need to change that. And the problem with this pressing and unmet medical need is that it's underfunded. So I'm going to throw some numbers up here, and please, for the experts in the audience, correct me. I'll share my sources with you, how I got to these numbers, and in case I'm a little bit off. But I think the point of what I'm going to share with you, regardless if the numbers need to be adjusted, would be only slightly, really drives home the point that we do not have the funding and resources we need to solve this problem. And this was really brought home to me while visiting the Florence, Italy for the ISPID conference, when Kyron put up some very similar slides. So I went back and tried to reproduce what he had put together. And what he did was make a comparison between sudden unexpected infant death and pediatric cancer. These are both illnesses with very terrible outcomes. And so I don't mean to put one above the other, but I think it's important for us to look at these two side by side for some perspective as in terms of a medical need that's not being met in the United States. So when I was looking through the databases, I was looking at pediatric cancer from 1 to 14 years of age, which has a population depending, an average annual population, depending on which database you're using, anywhere from 40 to 45 million or even 49 million. But we'll go with 40 million to say this. And while there are 15,000 cases of pediatric cancer per year, only about 1,100 of those on an annual basis end in death. And I think this is a testament to all the work we put into understanding and beating cancer. But it is not a solved problem. Nonetheless, annually, the National Cancer Institute puts forward over $250 million in research. I know Texas alone has a multi-billion program for cancer research, much of which goes to pediatric cancer. And this doesn't count on all of the different foundations that drive research into pediatric cancer. So look at SIDS. And if we take what we are looking at is a lifespan of 11 months. So that represents an annual population of about 3.3 million infants in the United States. And when we look at SIDS, or a conservative estimate of sudden unexpected infant death with no associated intrinsic extrinsic factors, we have a conservative estimate of about 1,400 infants. And I can't tell you how much NIH spends on solving this problem. When I go to the NIH databases, this is called NIH Reporter, and I search for grants that have the keywords sudden infant death syndrome in them, there's less than 60. I think there was 59 when I looked a couple of weeks ago. 5 of them come out of my own lab. I'm not trying to be braggadocious to say that 3 of those are fellowships from my trainees, and I'm very proud of their hard work, and they've accomplished that. This is really an underfunded, overwhelming, unmet medical need. And so when we look at this, we have 1 in 40,000 children in this age range are going to succumb to cancer. When we look at the conservative definition of SIDS, this is 1 in 200 or 26 infants. And if we include SUID, then we're looking at a lot higher rate. So in addition to this, we have an overwhelming need. It's underfunded. And then in my mind, this is one of the most difficult problems to solve in biomedical research today, not just because SIDS is mechanistically complex, which it is, but what we are dealing with in the most difficult cases is the death of an otherwise healthy infant that after an autopsy, death scene investigation, and medical history review, you have to go back to the parents and tell them, we don't know why. And as a scientist, that's also a very difficult place to start. It is diagnosed after the fact. So unlike other terminal illnesses, you have the opportunity to see the disease progress, and more importantly, you have the opportunity to intervene and see if that can make a difference, right? You gain mechanistic insight from that. That is taken away from us as clinicians and scientists to understand this. We have no predictive markers for SIDS, so you can't, sorry, I got that mixed up. There are no predictive markers for SIDS, and so that's where you lack the ability to intervene and gain mechanistic insight. Given that it's diagnosed after the fact, what we don't know in almost every case, with very few that have been recorded, and that's happening more frequently with at-home monitoring, is really what are the sequence of events that lead to sudden infant death and other sudden unexpected deaths in pediatrics. We have some important clues from a few critical recordings that have been made, but on an individual basis, we really don't know what the process was that that infant or child went through as it passed away. And that could be invaluable both in adjudication and for us to understand from a research perspective how we might intervene. And then another thing that we have been dealing with is that the sudden and unexpected nature of SIDS means that it's captured by the medical legal system. And this creates a gap. In other pediatric terminal illnesses, these are handled with ongoing care with a physician in a hospital that often has a research program, and there's a natural connection between the research field and the clinical care side. This happened, what we have here is this large gap, and it's a gap we're working to close at the center, and a gap that Savannah, as you've seen through her efforts, is working diligently for us to bridge here in Houston through her education work with law enforcement and engaging the medical examiners across Texas. So in my mind, these limitations hamper research into this problem like no other disease. It's not all bad news, right? We've seen a lot of great work that has led to a lot of our understandings of what potential mechanisms are in SIDS. And they come from a variety of studies across epidemiology, omics, and as we've seen from Hannah and have honored her for, careful neuroanatomical studies that give us great detail. And she has been involved in all of those aspects of our understanding of sudden infant death syndrome. So what this leads us to is my interpretation of the triple risk model, the multimodal risk threshold, which is that every infant is going to be the result of several different converging risk factors that we've all discussed. But the problem is, for every infant, the severity of both the intrinsic and extrinsic risk factors will vary greatly from case to case. So our greatest understanding of this coming from those risk factors, to extend that and to really understand and get us to the next level, we need increased studies that offer insights that give us mechanistic understanding of really what's going on. So what does cigarette smoke really do at the cellular and molecular levels to stop that infant from breathing or its heart from beating? So we have these SIDS-associated factors, and really the underlying physiology in much of this at the cellular and molecular levels, for the most part, is a black box. And how then those cellular and molecular changes then impinge upon cardiorespiratory physiology is a black box, and we end with a lost infant. So solutions for ending SIDS will require, and this has been said many times already, a reliable way to identify vulnerable infants. And once we can identify vulnerable infants at the individual level, not at the population level, Then we need prophylactic and acutely applied therapeutics. And as with some of the data I'm going to show you today, the type of therapeutic that may work for one type of infant that's been identified could potentially be lethal for a different infant. So both identification and treatment of vulnerable infants will require clear mechanistic understanding of the risk factors and failures leading to SIDS. So coming back to our multimodal risk threshold for us to try and understand those different risks and how they can lead to a SIDS outcome, we've been focusing mostly on sleeping in a face down position, trying to probe the mechanisms that fail when a SIDS infant is found in the prone position. So current thinking is that when an infant is found sleeping in a prone position, as Hannah has already said and many others, that they are rebreathing and they're becoming increasingly hypercapnic and hypoxic. And brainstem centers sensing this will activate and engage two different pathways. First, they will try to arouse the infant and then start the infant to take deep breaths. And if that's successful, then the child will come out of that dangerous situation. But if there is a failure of arousal, rebreathing will continue to the point that the infant is hypoxic and hypercapnic enough that it falls into an apnea and into bradycardia or slowed heart rate. When that happens, there's a last line of defense, which is that the infant will take very deep gasps, hoping to bring oxygen into the lungs that will then immediately reoxygenate the heart and ultimately reoxygenate the brain parenchyma, resulting in, again, restored cardiorespiratory function. However, if that gasping fails to reoxygenate the heart, lungs, and brain parenchyma, then we end up with a sudden unexpected death in sleep. So what we need to better understand the mechanisms that can lead to this failure and outcome are approaches that allow us to recapitulate SIDS-SUID associated intrinsic and extrinsic biomarkers in an animal model that can then be used to observe the pathophysiology relevant to SIDS and SUID. So what we're trying to do is take these SIDS-associated factors, which in case of an infant, have a black box effect on their cardiorespiratory physiology, and use an animal model to see exactly what that factor and its impact is on the animal's cardiorespiratory physiology, and come out the other end with an understanding of how single and multiple factors in different combinations lead to death. For example, how neural and cardiorespiratory function are perturbed with each of those different components. And once we have those verified SIDS models, that provides us with a potential biomarker that can be taken back to clinical studies for validation. And we can continue to test those verified models for different interventions, as we heard from Pete as well, to develop possible acute or prophylactic treatments for those different identified SIDS at-risk individuals. So I'm going to tell you then about how we are modeling autoresuscitation, that process of rebreathing that we think infants go through in a SIDS case. So we start with a mouse pup at P7, and here you can see recordings. See my mouse, all right? Recordings of the breathing and the heart rate. While we're recording the mouse, we're going to expose it to a gas mixture that's 97% nitrogen and 3% CO2. And we're going to expose the mouse to that until they stop breathing, but their heart continues to beat at a very slow rate. And as soon as that happens, we switch the gas back out to room air so that when they take that first gasp, they get oxygen back into the lungs to reoxygenate the heart and the brain parenchyma. The animal will continue to gasp to continue to resuscitate, to reoxygenate, and the heart rate will come back up until they go back into normal breathing. We let them recover for a short period of time, and then we repeat this assay over and over until the animal succumbs. And one of our major outcome measures is how many cycles can this animal survive. We look at a lot of other factors that are highlighted on here, but for brevity today, I'm going to just focus on how many of these cycles our different animal models can survive. So the goal with this assay then is to develop a SIDS discovery pipeline where we can bring together all of those known extrinsic and intrinsic risk factors in different combinations, apply them to our animals to develop models through genetic engineering and different types of exposures, test their output through high throughput phenotyping, and then use rigorous data analysis to give us mechanistic understanding as to the different kinds of failures that happen in these models. So the way we do this is we use a highly automated assay for testing cardiorespiratory function in the mice. Typically, in the past, this has been done manually, where one observer could test maybe 2 to 3 animals a day. But we've automated this so that it's a hands-off approach. And what you see here is a robotic platform that is self-calibrating. And what it is doing is moving these bell housings around, where they're going to be used to apply different gases to a mouse that will be attached here to this pneumatac that records its breathing. We calibrate the pneumatac with an automated pipette so that we have a consistent measure of tidal volume. Now, this is my grad student Deepak, who's now going to attach this mouse to a face mask. Creating an airtight seal. And now that face mask is going to be attached to the pneumatac to record the animal's breathing. And we instrument the animal by adding ECG leads to record its heart rate. And this is just a warming chamber being placed over the mouse. And so what's happening here is the mouse is breathing, we're recording it here, and every breath is being marked by the computer as well as every heartbeat. It's now being exposed to that anoxic gas, so it's going to breathe faster and deeper, as you can see there. And the computer is looking for a cessation of breathing longer than 3 seconds. And once we hit that 3-second threshold, the computer in real time recognizes that and then switches the gas back to room air. Now, the animal is not dead, it's simply apneic and bradycardic. Now there's that first gasp trying to resuscitate. You'll notice then shortly thereafter, the heart rate interval will shorten. There's a second gasp, and the animal is on its way to recovery. What's nice about this is that it's completely automated. When the robots are working really well, you can walk away and it'll take care of all this work for you. And if you can build one robot and load one animal to give you that ability to automate your work, why not build 10 robots so that you can do 10 animals at the time you would do one? And with these 10 systems, we've assayed nearly 5,000 animals across a variety of different models. Now, that work, all that modeling, all those assays produces a huge amount of data. So we put together a data analysis pipeline called Breathe Easy, the development of which was led, of course, by Savannah in the laboratory, and that's one of her first author papers. And that allows us to go through, as you can see in this movie, and very quickly, without having to know a lot about coding or anything, bring in your data in the various files and allow it to be analyzed in an unbiased and consistent manner, because every time the computer is going to look at those waveforms the same way, rather than hoping you see something when you're the observer and you know your mouse might have a particular phenotype. So this is just walking through all the different features that we've put our time and effort into developing to make this both simple upfront to use, but to give you the flexibility and depth to fully interrogate the respiratory waveform. So for brevity, I'll just skip through that. But now newer versions are in development in the laboratory, again, in efforts led by Savannah, Chris, and others to both look at the respiratory waveform as well as ECG data and unique features within the autoresuscitation reflex. So coming back to our multimodal risk threshold, we do now have a good way to look at the effect of sleeping in the face down prone position such that it induces autoresuscitation. And we want to now combine that risk factor with models of different brainstem neuropathies. And of course, to keep in line with our theme today, I'm going to share some data with you about our work looking at the various serotonergic neuropathies that have been discussed by Hannah and Robin and many others today as well. And the first set of experiments I'm going to tell you about are a very unique set of experiments that I don't think have really been done anywhere else in the field, or even in very rarely done like this in the field of neuroscience overall. And so what Savannah has done is set up a system that I'm going to walk you through, where she's been able to not only delineate very specifically the serotonergic system, but a second neurotransmitter system within the brainstem, the noradrenergic system, and remotely go in and turn off one system or the other, or both at the same time in the same mouse. And not only can she turn one off or the other off or both off, but she can also turn them on. So she can turn all of them off, all of them on, or on off in different combinations. And the way she does this is by using a pharmacogenetic method that she helped develop through genetic engineering. And her paper is highlighted here. And in that paper, you can see all of the data that validates these different alleles. And basically what she has are two different alleles that use a DREAD. These are modified muscarinic receptors that are biologically inert. They're produced in the cell that you want to target, and they do nothing. But when you give the animal a small injection of a ligand called clozapine N oxide, or CNO, then depending on which DREAD, if I can find my mouse, sorry, is present in those particular cells that you're looking at, you'll either turn that cell down or you'll turn it up. And so these are called HM3D and HM4D. HM4D will turn the cell down, HM3D will turn the cell up. And spending more than 2 years just breeding these animals together after their development and through genetic engineering, she's come up with 8 different experimental groups. So first, she can go into, as I said, each system individually and turn it on or off. Then she can go into both systems and turn them both on or off. And then she can go into both systems and turn one on while turning the other one off. So this allows her to look at all the unique possible interactions between these two systems as they regulate the autoresuscitation reflex. Savannah is a very thorough person. So she made sure this experiment was robustly designed. So not only does she have all of these different groups of animals that she's testing, but she tests them in a controlled fashion with a placebo injection. She tests them at a low concentration of the activator and at a high concentration of the activator. She looks at males versus females. So then altogether, that creates 54 different groups of comparison. And in order to power that statistically, so you have a rigorous experiment, she's had to test nearly 900 animals for this experiment to be informative, which is made possible by robots. So here are the outcomes. One of the first things you can see here is that compared to our saline, sorry, I apologize because I can't see that. All right, so compared to saline, when we give the activator, it itself has an effect, and this is an important outcome that we must recognize and take into account. So all of the further comparisons of those different genetic groups that I'm going to tell you about are compared to that CNO, these CNO control groups here. Overall, when she turned on and off these different combinations of neurons, what she found is that 5 of the groups were vulnerable. And they largely consist of groups of animals where the noradrenergic output has been increased. And there's one unique group here where serotonergic output being increased creates also a vulnerability similar to increased noradrenergic output. And it doesn't matter with the noradrenergic neurons, whether serotonin is going up or down, the animal remains vulnerable. And you can see here, depending on how much of the activator, the severity of the vulnerability changes as well. So the more activator you give the animal, the more vulnerable it becomes. Interestingly, when Savannah inhibited the serotonergic neurons in this experiment and compared it to the CNO controls, we did not see a change in the outcomes. They did not show an increase in vulnerability. So the conclusions from this is that any combination of noradrenergic neuron stimulation and serotonin manipulation results in early lethality. Serotonin stimulation, surprisingly, also drives early lethality in our assays. And serotonin neuron inhibition, particularly when compared to the CNO controls, shows no differences. Additional considerations that we have to think about is that CNO itself has a major effect on the assay, but we do control for it. And we should be thinking about how acute perturbations mechanistically compare to the dysregulated but presumed steady state of a SIDS infant. And I think we also need to think about, looking at this data, what the synaptic consequences are of the different serotonergic neuropathies that we've discovered, or that the field has discovered over time. It's a very difficult and impossible, actually, I would say, in humans, problem to understand whether or not the serotonin levels you see in a tissue assay represent the serotonin levels inside the cell or at the synaptic cleft. So another model that we've developed in the lab as well is to look at the serotonin receptors themselves. So what Savannah's work has been is to simply, not simply, quite in a very complex way, go in, but cleanly delineate the different populations and turn on the activity of those cells. What Deepak is doing here in his work is looking at serotonin receptors, and particularly the 5-HT1B that Hannah had mentioned before. And what he wants to do here is understand what the role of the 5-HT1B receptor is in autoresuscitation. And when he removed the 5-HT1B gene from the mouse and tested it with it having either just one copy removed or both copies removed, what you see here is that these mice have a vulnerability. Where's mine? Start from here. So these are the wild type mice that have normal 1B receptor function. And what you see is a numerical trend. As these mice lose one copy, they show a potential vulnerability. And then certainly when you remove both copies, they do not perform well on our autoresuscitation assay. So future experiments that Deepak's engaged in is trying to understand what cells are important. 5-HTB is expressed in a wide variety of cells within the brainstem. So he wants to know which cells within the brainstem or elsewhere in the body are the critical ones where that 1B receptor function is essential for autoresuscitation. And another question he's asking is, can we rescue these animals that have a very strong vulnerability when they're missing this gene? And for that, he looked to caffeine. Caffeine is a common therapeutic used in the NICU for addressing respiratory deficits. As Hannah mentioned before as well, Kevin Cummings and others have used caffeine in similar autoresuscitation models, particularly here in this paper with the PET1 deficiency, showing that it indeed can rescue to a certain extent the autoresuscitation deficit of a mouse model that has perturbed serotonergic neuron function. And so what he was able to show is that in his experiments, the control animals that are lacking 1B function, but either did not get an injection or an injection of saline instead of caffeine, show that vulnerability that I just showed you earlier in those slides, that they don't resuscitate as well as the control animals. But when he brings up or adds caffeine to those mutants and tests their autoresuscitation reflex, that vulnerability disappears. So the next question out of this is, can we go bigger? We have 10 rigs, we have a software to assess everything, and we are. So we're developing the Center for Cardiorespiratory Physiomics with an investment from Baylor, where they built a room for us to add additional robots. So the centerpiece here is for adult systems, but on the walls here, we're developing additional robotic platforms that will allow us to test as many as 150 pups a day when completed. So the goal with building this is to develop a high throughput discovery pipeline, as I just laid out before, where we want to take those intrinsic and extrinsic factors that have been identified in the literature, engineer those into the mouse, either through external exposures or genetic engineering, test them through a high throughput approach using our phenotyping platforms, and then use our software for rigorous data interrogation to give us those critical mechanistic insights. But we want to take this one step further, and I think that's been clear as we've been developing this center, and you've seen from much of the work that Savannah has been doing, we don't want to just take from the field what they are finding and test it, but we want to also find our own factors to test. And that has been the goal of the Isabel Davis Center. So we are already working with others to develop a human full genome sequencing program here. And you heard about Savannah's epidemiology work, finding that mold signature that she's working to test here in the near future. With our genetic engineering, there isn't a variant that we can't put into the mouse so long as some basic rules are met in terms of orthology. And we will soon have the capacity to test almost all the models out there that we can build as fast as we can. And now that we have a consistent way to interrogate those waveforms instead of using hand annotation, we can now begin to apply machine learning to those outcome measures for additional insights. So with a front to back pipeline to gain mechanistic insights that will lead to prognostic indicators and prophylactic therapeutics, we really hope that we can make a difference. So as Hannah mentioned, her first grant was called a fishing expedition, and I've had that one thrown at me so many times. And so I just decided to own it. Do we fish? Yes, yes we do. So, since I'm the last speaker and I'm already making everyone wait past dinner time, I'm going to take just one last moment to say thank you to a number of people. So first I want to start with my lab overall, both for the work I've showed you today. There's been so many people who have made this possible, but also my lab has really come together to make this conference a success. And I'm very grateful to everyone pitching in, running tables, moving stuff around, handling my last minute emails saying, help me and let's do this. They've been unquestionably committed to this, and I just want to say thank you to them right now as well. Of course, we have to say thank you to Savannah for all her hard work on the conference. She really was the driver for all of this, the organizer, everything, and you know that. And then the last thing I want to say about Savannah is that I've really tried to make it clear from the beginning that she has been an essential 50-50 partner in starting and growing the Isabel Davis Center to what you see here and now. And for that center to continue into the future,